Good evening, Samish. Okay. So, did you go through the video of the last lecture, or shall I repeat it for you? The last lecture video upon which I took regarding the polarization of light. Did you go through the video? Did you understand, or shall I repeat, or do you have any doubts regarding that video? Okay. So, shall I repeat the polarization part? And just give you a gist of the polarization part. Okay. Good evening, Pratham. Pratham, do you have any queries regarding the polarization part, or shall I repeat little bit? Okay. So I'll just give you a zest still so that you are in the frame of mind, otherwise it will be too crude to start. Okay, so I'm Rahul Saha. Today we'll be talking about the Haworth projection, Fischer projection. Just I'll give you what is polarized light, a zest of it. Okay. So simply if you say, if we take only two dimensions, okay. So normal light, so there is something known as stereochemistry. Okay. Means the isotopes which are, you can say, they will bend the polarized light in one way or the other. Okay, so first of all we'll talk about what is polarized light. Okay, so what happens is, the most common method of polarization involves the use of a specific filter known as a Polaroid filter. Okay, Polaroid filter. So you can purchase it anywhere. Okay, so they are made up of special material which is capable of blocking, you can say, one of the two planes of vibration of an electromagnetic wave. So we know that an electromagnetic wave has both, you can say, particle and wave theory, quantum and wave. So it has dual nature. Okay, so if you consider the wave nature, okay, so in this sense, a Polaroid serves as a device that filters out one half of the vibrations upon transmission of the light through the filter. So suppose this is the Polaroid filter. Okay, this is the Polaroid filter. This is the axis of the Polaroid filter. Polaroid filter. Okay. Now you have unpolarized light. So I'm only taking two axes here, not making it three. It will make it complicated. So it is vibrating in the y-axis and x-axis. Okay. So after it passes through the Polaroid filter, okay, what happens is, the light that emerges this side, okay, it will emerge with one half of the intensity and one of the way of the vibrations. Means vibrations is in, or oscillations in, one of the axis, okay, which must be in a single plane, and that light is known as a polarized light. So suppose it is coming out as a light like this. Okay, so a Polaroid filter is able to polarize light because of the chemical composition of the filter material. Okay, the filter can be thought of as having long chain molecules that are aligned with the filter in the same direction. Okay, so what happens is, during the fabrication of the filter, the long chain molecules are stretched across the filter so that each molecule, as much as possible, is aligned in, you can say, the vertical direction. So as unpolarized light strikes the filter, the portion of the waves vibrating in the vertical direction, okay, are, you can say, absorbed by the filter. Okay, so the general rule is that the electromagnetic vibrations that are in the direction parallel to the alignment of the molecules will be absorbed. Okay, so what you will get is you will get this light. Okay. Suppose this is the axis, this is the axis. Okay. So this one will be absorbed, okay, y1, and what you will get is this as the polarized light, only having vibration in x-axis. Okay. This is the polarized light, and you have certain angle also at which this light hits that to this specific thing. That is your theta b or Brewster's angle, that will come in physics in class 12th. Okay, so we will not go in that deep physics here. So just the thing that this is a polaroid meter. You have light, x and y vibration. If the molecular axis present in the polaroid filter is in y-axis, then y-axis will be absorbed, intensity will be halved, also vibration will remain in one axis, and the light that vibrates is in only one axis is known as a polarized light. Okay, that is simply a polarized light. Okay, this is the concept of polarized light in short. Okay, so yeah, so that is what I gave you the recap. Okay, so this is a simple polarized light. Okay, so now why do we need polarized light? Okay, why do we need polarized light? The thing is, there are molecules in chemistry or in biology also which are simply known as the chiral molecule or achiral molecule, meaning that if you take any amino acid, okay, let's take an amino acid. The best example would be to take any amino acid. An amino acid, you must have read in biomolecule chapter, looks like this. Okay, suppose this is your alanine. This is your alanine. This is how your alanine looks. This one is your you can say glycine. This is alanine, this is glycine. Now, what is the main difference between an alanine molecule and a glycine molecule? What do you see? The main problem or the main difference between alanine and glycine? So, first of all, we should consider this COH as the main functional group. Okay. So, the first carbon attached is alpha carbon, C alpha. This is the central carbon. Okay. Now, look at this central carbon. The central carbon, if you look at, okay, you can say, look at this central carbon, and there are 4 different groups attached to this central carbon, specific one. This is different, CS3, NH2 is different, H is different, COH is different. So all 4 groups are different. Here if you see, all 4 groups are the same. Sorry, all 4 groups are not different here. All 4 groups are not different here, because this and this are, these two are the same groups, only these two differ. Okay, so the thing is, when will I call a molecule or a compound chiral? When it will be having a chiral center. Okay, chiral means CHIRAL. Okay, chiral molecule will be your alanine will be chiral, this will be achiral. So this is a very important neat question. Which amino acid is achiral in nature? So only one amino acid is achiral, that is glycine, the smallest amino acid. Because to the alpha carbon, to the central carbon, all the 4 groups attached are not different. Okay, the 2 are same, H and H are same. Any other amino acid, if you look at, to the central carbon, the 4 groups attached will be different. All the different groups. CS3 is different, COH is different, H is different, H2N is different. So all 4 different groups. Now, what does chiral mean? Okay, what does chiral mean? So chiral mean that the meaning of chiral is, now coming to chiral and achiral. Why did we do all this? Why am I calling it chiral? Why am I calling it achiral? Okay, so if you pass a plain polarized light, okay, let's draw these 2 molecules. CS3, this is alanine, one if I draw, this is glycine. Okay. Now let's pass the polarized light, not a normal light. Normal light we do not use because it will not show any kind of result that I want you to see. Getting? So I have first converted normal light to polarized light. Now I'll pass the polarized light. Okay, suppose this is the polarized light passing through. I've used a Polaroid filter, converted normal light into polarized light which vibrates in x-axis. It is passing through this one. Now, it will, if it is a chiral molecule, because C alpha, all the four groups are different, so alanine is a chiral molecule. So if a polarized light passes through a chiral molecule, okay, it will not pass in the same direction. It will either rotate in the left or the right direction. If it rotates in the right direction, this molecule will be known as a D isomer or dextroisomer. Dextro or D means right. If the polarized light rotates in the L direction, it will be a levo or L isomer. So depending on D or L, it will become either a D or L. I'll show you how it is D and L. But first know that chiral molecule, if it is present, if a polarized light passes through it, it will either be rotated in right or left direction. It won't pass through in the same direction. It won't emerge in the same direction as the direction at which it entered the molecule. Whereas if the same polarized light PR having vibration in only the x-axis is passed through glycine, which is an achiral molecule, because two groups are same, all four groups are not different. It will pass through in the same direction. It will not be able to rotate because this molecule cannot rotate the plane polarized light in either left or right. Okay, that is why this molecule is achiral. It will not generate stereoisomers. Okay, here it is generating stereoisomers. Now how will you say whether it is D or L? Okay, whether it is a D alanine or L alanine. Now the question arises, sir, how will I say, or how will I know that alanine will convert this light in, or will divert this light in right direction or left direction? There is again rule for that also. That rule is known as Corn rule. C-O-R-N, Corn. Corn is nothing but CO stands for COH. This is the R group of the amino acid. Okay, this is the NH2 group of the amino acid. So you are supposed to go from COH to R to amino group. Okay. So the Corn law is the rule that allows to distinguish enantiomers from each other. Okay, I'll tell you what enantiomers are. Okay. In other words, it determines simply the amino acid, whether the amino acid is a L isomer or D isomer. Okay. So we all know that every amino acid is like this. At its core, it is like this. This is the core. This is the R group. In glycine, the R group is H. Alanine, the R group is CS3. Okay, every amino acid differs here only. So COH to R to H2N. This is how we will go. Okay. Now, this is the core of any amino acid. Okay, any amino acid will have these three. Okay, groups attached to the alpha carbon. Okay, central chiral center. So carboxylic acid group, amino group, and a hydrogen atom, and it will have a distinctive R group, okay, which is the representation of the side chain. So the Corn is an acronym I told you for COH of the R group followed by the COH of the central carbon followed by the R group and the amino group. Okay, so if you have now apply Corn rule to these two molecules. Simply, this is alanine. Okay. Now this can also be alanine. This can also be your alanine. Okay, this is also alanine. Now, which will rotate the light in right, which will rotate the light in left direction? Okay, alpha carbon. That we have to determine. Now, if we apply Kohn rule, okay. Now, if the carboxylic acid is followed by the R group and the amino group in a clockwise direction, okay, in a clockwise direction, then the amino acid is an L isomer. So carboxylic acid, this is the R group, okay, this is NH2. Which direction is it? It is clockwise. So I can call it L-alanine, means when the light will pass through this, it will be rotated in the left direction. Okay. Now carboxylic group, this is the R group, this is the H2N group, this is which direction? Anti-clockwise. So when the light will pass through it, it will be rotated in the right hand side. This will be your D-alanine. Okay, so think of how close or how intricate the isomerism is. Just this side or this side, you have H2N and COH, and these two molecules will indeed experimentally rotate the light in opposite directions, or they have been found to experimentally rotate the light in opposite direction. By looking at this concept only, this rule has been formulated. So this is an experimentally tested rule known as the Kohn rule. Very, very important rule for both biology and chemistry. Organic chemistry later on, and biomolecules or biochemistry. Okay. So all the amino acids in our body are L-alanine. Always remember. Again, neat question. All the amino acids are L-alanine, means that none of our enzymes are able to recognize the D form of the amino acid. Now think of how great our enzymes are at recognition, because both the molecules are same, but enzymes can recognize the stereochemistry of the molecule also. Think that if you have NH2 attached to this side only, then only L-alanine will be recognized by the enzyme, not this side. Even though the chemical formula, the structure is completely same. It is not even that you are making butane plus isobutane, that you are straight chain and branch chain. Both are same molecule, same type of chaining is there. Okay, no branching is there right now. Okay, so same type of configuration, same type of valency, you can say distribution. It's still due to the stereoisomerism, only L1 will be preferred, or is the only choice for the enzymes present in human body or any body. Some exceptions are there in bacterial cell wall. Some D amino acids are found. Okay, and that too in bacterial cell wall. Okay, that is an exception. But in human beings, bacterial cell wall means peptidoglycan layer. But in human beings, not or any other eukaryote, not a single amino acid, if you take it in D form, okay, the whey protein or all the amino acids present in the meat or all, they are present in L form because they are used in L form. So we take amino acids only in L form. Okay, the cheap amino acids which are sold in or at Amazon, they may be D amino acids because they are more expensive too. Because once you convert, you may get half D, half L. So if you want to filter it, they will be more expensive to make. Okay, so that is the thing here. Okay, so always think how great your body is biologically. Okay, because you have, what you have is L amino acid at your disposal always. Okay, so any doubt till here in the recap portion? Any doubt? Ramesh Pratham, regarding amino acid, regarding polarization, regarding stereochemistry. Okay, so I think this must be clear. So now, one thing I said was enantiomer. Okay, so enantiomer, I just tell you what are enantiomers. Enantiomers are this only. Okay, so enantiomer, okay, comes in biochemistry a lot of times in NEET question also. So enantiomer is nothing but it, you can say, look at these two molecules only. If you look at these two molecules, okay, so enantiomers are what? They are mirror images, okay, but non-superimposable. They are mirror images. You can say D-alanine and L-alanine. This one will be, you can say COH, this side will be D. This one will be your L-alanine. So they are mirror images, okay, but they are non-superimposable. Superimposable means if you keep these two on top of each other, they will not be able to superimpose or completely cover each other. Okay, you can try this at home by using 3D diagrams, or you can have some YouTube videos where 3D diagrams are present. Just type enantiomer and you will understand what is the meaning of non-superimposable. They are mirror images, but if you combine it together, if you post one on top of the other, if you cover one with the other, complete coverage, or you can say complete superimposition, okay, it will not be able to completely cover that part. Okay, that is known as non-superimposable enantiomer. Okay, one more thing I'll tell you is, it's a very good example of, you can say, pharmacology. Once you become a doctor, you will treat patients by your knowledge of, you can say, pharmacology and pharmaceutical or therapeutical drugs. Okay, so if you have a mixture, okay, in a tube, if I have added 10 ml of L-alanine and I have added 10 ml of D-alanine, okay, now I am passing a plain polarized light. Okay, so after it comes out of the tube, okay, it comes out of the tube, which direction will it take? Will it take this direction, the right direction, or left direction? Which direction will it take? Just understand the question. I have added in a tube 10 ml of L, 10 ml of D, equal amount of both the, you can say, enantiomers or stereoisomers. I can say, I can call it enantiomer because they are mirror images but not superimposable, or simply I can call them stereoisomers / enantiomers. Now, which direction will they take? Okay. So the thing is, they will take which direction? They will take no direction. They'll come out as it is, because the net effect is getting 0 here, because this 10 ml L-alanine will try to move it at the towards the L side, 10 ml D will try to move it towards the D side. Okay, now what will happen is, both will cancel out and the net effect will be, they'll come out as it is. So you will feel like this one does not have any kind of chiral compounds, but it did have, but in equal concentration. So this kind of mixture where equal concentration of L and D enantiomers are present are known as racemic mixture, and the process is known as racemization. Okay, so the levocetirizine that we take, okay, if you see the second generation antihistamine drug, was cetirizine. Okay, now the third generation one has less drowsy causing effect, or doesn't make you sleepy. Okay, this used to make you sleepy. If you take this, you will feel sleepy. But if you take levocetirizine, that doesn't make you sleepy. No sleepiness, no drowsiness. Why? Because cetirizine was a combination of D-cetirizine plus L-cetrizine, both the stereoisomers. But levocetrizine means scientists have eliminated the D form. Okay, they have only concentrated on L-cetrizine molecule to minimize the side effect, because they found that L-cetrizine has most pharmaceutical value, most therapeutic effect, and doesn't make you sleepy. So this is also extremely applicable in pharmaceutical industry, this concept of, you can say, stereoisomerism. Most drugs are either L isomer or D isomer, like that. Okay. So now coming to the carbohydrates. When we study about carbohydrate, the basic molecule that we study about is, number one is the glyceraldehyde. Okay. So glyceraldehyde, if you talk about, it looks like this. This has a specific aldehyde group, that is glyceraldehyde, then C OH H, then CH2OH. This is your D-glyceraldehyde. And this will be your L-glyceraldehyde. This is your L-glyceraldehyde. Okay, so you can see this molecule. This is the aldehyde group. Okay, they are also known as hemiacetal. If you have studied organic chemistry, you will know this is a hemiacetal group. But when they combine together, these two, if it combines together, it gets further oxidized. It becomes hemiacetal, or you can say simply acetal, from hemiacetal to acetal. Or if they have ketone group, they'll be known as hemiketal. If they're further oxidized, they'll be known as simply ketal. Okay, so this is the Fischer projection. So if we talk about Fischer projection of glucose molecule, okay, so this is a how many carbon molecule? 3 carbon molecule. So if you take Fischer projection of a 4 carbon molecule, okay, like erythrose, if I take, okay, carbon number 1, okay, carbon number 2, okay, then carbon number 3, then carbon number 4. Okay, this is D-erythrose. Now why am I drawing D always, not L? Because it is just opposite. Again, neat question. Carbohydrates in the body are accepted as D isomers, whereas amino acids, they are accepted as L isomers in the human body. Okay, always know this for a fact, D isomer and L isomer. So that is why when I am drawing carbohydrates, okay, I am writing always first the D form, not the L form. Okay, so this is D-erythrose, a 4 carbon, carbon 1, carbon 2, carbon 3, carbon 4, 4 carbon carbohydrate. If we come to the 5 carbon carbohydrate, okay, the ribose, which you used to see in DNA, okay. So the ribose, if you see D-ribose, okay, it looks like this. H, this side H, 1, 2, 3, 4. This is 5. 1, 2, 3, 4, 5 carbon. This is your D-ribose, which is present in, you can say, DNA, but the oxygen at the second carbon will not be present. Okay, that is deoxy, but ribose is the constituent of RNA and deoxyribose is in DNA. Okay, so this is your 5 carbon carbohydrate. Now coming to the 6 carbon, the most famous glucose. 6 carbon formula is C6H12O6. So when you come to the 6 carbon compound, so you have glucose, mannose, galactose, okay, you have talose, many are there. Okay, so if you think about glucose only. So glucose looks like this. But third carbon, this side will be H, this side will be OH. One, two, three, four, 5, 6. 1, 2, 3, 4, 5, 6. You can count, it will be C6H12O6. This is your D-glucose. Okay, that is the meaning of D-glucose. Okay. So once you have, this is the Fischer projection, the straight line projection, or you can say linear projection of any carbohydrate is known as Fischer projection. Okay, this projection shows what? That you have a specific aldehyde group. In the end, you have CH2OH. Okay, and the arrangement. Okay, this is what it shows. It also shows one more thing that I'll teach you, whether you are an anomer or epimer. Okay, anomer or epimer, that also it shows. Okay, so if we talk about epimer, okay, epimers are very easy to comprehend. Okay, I'll just draw some molecules. So let's first talk about our epimer, and then later on we'll be talking about anomer. Okay, so let's talk about glucose, okay, and galactose. Let's talk about glucose and galactose. I'll draw both the molecules and you will tell me the difference. This is your one, two, three, four, five, six. This is D-glucose. Now, second carbon. This is known as D-galactose. Both have 6 carbons. Where do you see the difference? Carbon number 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Where do you see the difference? I can see the difference in, you can say, carbon number 4. Okay, carbon number 4, you have OH this side here. Okay, OH is in this side. Here in case of carbon number 4, OH is on this side, opposite side. So if you have opposite placement of the molecules attached to the central carbon is this one here, at any other carbon, okay, except the anomeric carbon. Anomeric carbon is always the carbon to which the the aldehyde or ketone or any functional group is attached. So to this carbon you have functional group aldehyde attached. Okay. So to the carbon to which the functional group is attached is known as anomeric carbon. So except anomeric carbon, okay, to which the functional group is attached, any other carbon, if the attachment of the groups, they are not in the same direction, they are in opposite direction, that is known as epimer. Okay. So I can say that galactose, D-galactose and D-glucose are epimers at carbon number 4, because at carbon number 4 OH is this side and for galactose OH is this side. And I'll also tell you what is the significance of OH being in this side or OH being in this side and H also. Okay, means what do you mean by this side and this side? How do you discern this significance? Okay, that will also come into picture. That is why I am making epimerization, or epimerization exists. If you can attach it in any direction and it wouldn't matter, then we wouldn't be calling it epimer. Okay, since OH is in opposite direction to the OH present in glucose at carbon number 4, rest all are in the same direction. You can see rest all H and OH are in the same direction, both. Here H is in opposite direction, H is in this direction, H is in the opposite direction towards that direction. So this is known as epimer. Okay, at C4, D-glucose and D-galactose are known as epimers. Okay. Now, once epimerization has been done, anomer I'll show you how it is done. Now let's convert Fischer projection to Haworth projection. Then everything will become clear. What is the significance of OH this side, H this side, H this side, OH this side? Everything will become completely crystal clear. Okay. So the thing is, first let's rotate. Let's draw a specific glucose molecule only, and let's show you what do I mean. Okay. So I can write this CHO group, CHO. Okay, the first carbon one in short. Now if I rotate this 90 degree here and here, rotate it 90 degree, what will I get? Okay. 90 degree rotation. Okay, that's what I am doing. So what will I get? It will become horizontal molecule. Once it has become horizontal molecule, it will look like this. First carbon is COH, second carbon, H is now downward. See, downward, upward, downward, upward, third, fourth carbon downward, fifth carbon downward. Okay. Now sixth carbon is CH2OH. This is the sixth carbon. So the meaning of this side and this side is, if you have OH on this side, that will be anything on this side. Okay, anything on this side will be downwards. You can look here. After rotation of 90 degree, anything on this side will be upwards. After rotation of 90 degree, you can see here, everything came upward here, everything came downward here. Okay, you can see like this. Look here, everything which was this side came downward, everything which was this side came upwards. Okay, now after this event, what happens is we will rotate. Okay, now again I'll draw this to avoid confusion. Okay, so let's take each of these molecules as a separate group. So if I take this one C5, okay, C5 carbon, if you see this side, it is this group is attached, this side H, this side OH, this side this group is attached. So C5 I can say is a separate chiral center or not. Just look, this entire group is different. Okay, this group is different, this group is different, and this entire group is different. Okay, so this is a chiral center carbon, C5. Independently it is a chiral center carbon also. Okay, now if you cause rotation, okay, if you cause rotation, okay, you rotate this like this and just rotate the molecules from its position. Okay, you cannot break this backbone, so H will come this side, OH will come this side, and this will come this side. This will not be rotated because this is a complete backbone. Okay, so H will directly come this side, OH will come this side, and this group will come this side. Okay, that's why I have drawn this and not ending here. This arrow ends here. So once we rotate it like this, what we will get is we will get this H came this side. Okay, here it was OH, it went this side. Okay, and CH2OH went here. Okay, now it went here. Now look what happens. The O of this one, this O has got, you can say, two lone pairs. Now this O and this C is kind of positive, partially positive. This O is negative. This O is highly negative because it is highly electronegative compared to H. So this will cause a nucleophilic attack. Nucleophile means nucleus loving, means proton loving or positive loving. So this will cause a nucleophilic attack on the first C of the, you can say, aldehyde group. And when it does a nucleophilic attack, what will happen is this thing will be connected. How? Like this. This bond will be broken. This bond broken. Okay. Now you have O here. This O will get attached. Okay. To directly, you can say, this thing, first carbon. Okay, and that H which was left from there, which got removed from there, it will get attached here. So now you have cyclized the chain. How many carbons are there? 1, 2, 3, 4, 5 and 1 O. So it's a 6-membered carbon chain. Now how will you be writing it? You will be writing it like this. Only O will be written separately, rest you have carbon, so we don't write carbons as a rule. Okay, at each center this is the first carbon. Okay, this is the second carbon, third carbon, fourth carbon, this is the fifth carbon. Okay, 1, 2, 3, 4, 5, carbon number 1, 2, 3, 4, 5. Okay, so we don't write that. Sixth one is the O, which completes the ring. Now active first carbon, OH is up. That is the meaning of this side or that side. H is down, so H will be here, down. Second carbon, OH is down. H is up. Third carbon, H is down. That is why H that side. That is the significance, down or up. Okay. Then fourth carbon, OH, down. H up. Fifth carbon, H, down, which is up. CH2OH. So this is known as your Haworth projection. This diagram you will be looking at your book. This also you will be having in your book. This is how to convert Fischer from Haworth. That comes in PMT examination. Okay. So they will give you the Fischer projection. You will be given 4 Haworth projections, which is the correct answer. Like those questions will also be asked in biomolecule chapter. The biomolecule becomes very, very tough, that's why. And if you know the basics that I have taught you, this basic you will not find in any reference book also. They'll directly convert without, they'll just say nucleophilic attack. So this is my personal finding, which that I have done to simplify it, okay, converting it into vertical direction, from vertical to horizontal, and then showing the entire linkage. So if you stick to this, you can convert any Haworth to Fisher and Fisher to Haworth also. You can convert it anti in the opposite direction also. Now coming to, you can say, now tell me if you have any doubt here. Okay, any doubt here. No, don't need to remember the structure. Only you need to remember the glucose one that I made you see. Only this one. Only this one you should know. That's all. No need to remember any other structure. Only glucose you should be knowing. At least. At least glucose, if you know, you will be well served in the PMT examination. Just know the trick how to convert Fisher to Haworth. Okay. Why don't you write a book? Writing a book will be a daunting task, Samesh. Like, many people have asked me to write a book, but again, finding a, you can say, publisher, then getting that book copied, it needs a lot of, you can say, investment also, right? Investment plus searching, writing, copyright, this, that, so many things are there. I taught you copyright and trademark, right? I have to get that thing also done then, so that nobody can steal my stuff. Okay, so that's why I've simplified it for you. This lecture will be in wisdom always. Okay, so this thing has been simplified. In your reference books, you will not find this simplified thing. They'll directly go from Fisher to Haworth, directly jump upon it. Okay, but this is the simplified version and logical version of how to convert. And this also serves the purpose, what is down, what is up. Why you have this side and this side, that completely says OH this side means down and up. Okay, directly it will solve the purpose. By naked eye only, OH is up, so here I have kept OH up. H is down, here I have kept H down. Okay, so now coming to the next part known as anomers. Okay, if you come to anomer, okay, anomer means if the OH is on top in the, you can say, first group or on the first carbon, which is the anomeric carbon I told you, because this has or this had the hemiacetal carbon. The first carbon, if you remember, this was like this. So if the OH is on top here, this is known as beta D glucose. This will also be in your book. Beta D glucose, alpha D glucose. If at all this OH comes down, H comes on top, that can also happen. So I'll draw the image here. So H on top, OH down. If this happens on the first carbon, which is the anomeric carbon, okay, then H, OH, OH, H down, and again H up, OH down. Okay, if this is what it happens, okay, that is known as alpha D glucose. This is known as anomer. Alpha and beta are anomers. Okay, because they are happening on the C1 carbon, which contained the functional group. Okay, simply functional group. In case of glucose, it was an aldehyde group. Okay. If their arrangement becomes opposite, okay, then they will be known as beta or alpha. So in your book you will be having alpha D glucose, beta D glucose. Even in NCERT it is given alpha D glucose, beta D glucose, but the reasoning is not given, and that same reasoning is actually NEET examination. Okay, so that is how you prepare for NEET. Okay, everything which is given in the book has to be reasoned. Okay, the reasoning has to be learnt. Okay, so this is alpha beta, very simple. But the thing is, first carbon you must look at, or any carbon at your level, first carbon, but at higher levels, any carbon which have the kind of functional group present. But when it comes to glucose, it is the first carbon only. So for simplicity, first carbon, if it is just the opposite sense, it will become beta, it is alpha. Both are D glucoses. Okay, that is your anomer, that is epimer. Okay. Yeah, sure, Pratham. Don't worry. Whatever I am teaching, okay, just concentrate on that. Every neat question where I'll send the, you can say, past paper to you. If you have learned my lectures completely, you can solve any question of NEET from the past papers. Okay, because those papers from 1998 to 2019, we have compiled the past papers of every chapter. So we will deal with the biomolecule part. If you learn all these things, those questions will be very, very easy. Biomolecule is a very, very, you can say, a logical and rational chapter. There is no need to mug up a single thing here. It goes directly like that. Okay. Yeah, I know it's a lot to comprehend. That's why I'm saying just listen to the lecture. Don't even open the book. Okay, download this lecture and listen to it. Don't even open the book. You can solve any question. Okay, that is what I'm saying. If you keep on opening the book, you will be a lot, you can say, confused. Okay, because these things, if you learn from a book, Polaroid, polarimeter, you will see huge notes. Okay, and rote learning is like, I usually prohibit because it burdens the students a lot. That's why short notes like this, if you want, I can provide you short notes also. Because in Allen also, you can see one Shoaib was there. He scored full marks. Okay, so he was an Allen coaching student. Same thing he learned from the notes. Okay, so if you learn from the notes, if you have the concepts, okay, that he had concepts, he scored full. So scoring full is not a great deal. Okay, even one girl was there, Anshika. So these two have, for the first time, they have scored 720 out of 720 in NEET examination. So it is not that difficult. Okay, it's quite easy, provided you know the concept. And not only in biology, you should know the concepts of physics and chemistry also, right? Because physics chemistry also is asked in length or at length in NEET examination. So now we may come to the disaccharide. Okay, disaccharide. And today we'll be completing the carbohydrates. Just we can come to the disaccharide structure. Now, if we come to disaccharide, I'll tell you two things: reducing and non-reducing sugar. Okay, very easy to comprehend, reducing and non-reducing sugar. Okay, so these two things are also asked in NEET examination. What is reducing sugar? What is non-reducing sugar? Okay, so the thing is, in reducing sugar, what happens is the first carbon of one of the agents remains, or one of the monosaccharide remain unbounded. I'll just show you. Okay, suppose let's take alpha D glucose and beta D glucose. Let's take these two molecules. Okay. HOH. This I am drawing alpha 1. Okay. Then HH. Then again you have HOH. Then HCH2H. This is your alpha D glucose. Now this is your simple one which I had shown you in the beginning, beta D. OH, H, H, OH. This side H downward, rest again, H upward, again H downward, CH2OH. This is beta D glucose. Now this is the first carbon. This is the first carbon, second, third, this is the fourth carbon, fifth carbon, sixth carbon, second, third, fourth, fifth, sixth carbon. Now, if these two glucoses they form a bond, because monosaccharide will form disaccharide, disaccharide will form polysaccharide, just like amino acids. So the bond produced is again a covalent bond that is known as glycosidic linkage or glycosidic bond. Means if the bond forms, this H2O will be released out, condensation reaction. Okay, after this H2O is released, the O will join. Okay, so like this it will be joined. So this O will join like this. So this is alpha 1 first carbon to fourth carbon 1,4 glycosidic bond or glycosidic linkage you can call bond. So this is how disaccharides are connected. But this one is a reducing sugar. How? Because the first carbon of one of these disaccharides is still vacant, and this is the carbon which had the functional group. Okay, so this carbon is vacant, this is not attached. First carbon of this one is attached to the fourth carbon. So of this beta D glucose, the first carbon is vacant. Of alpha D glucose, first carbon is occupied, or it has participated already in the glycosidic bond. But the first carbon of beta D glucose has not participated. It is still vacant. So if the first carbon where the hemiacetylation was there, or the hemiacetal carbon or the anomeric carbon of one of the, you can say, monosaccharides present in a polysaccharide or a disaccharide is vacant, that is known as a reducing sugar. Okay, and if the configuration was like this, if the configuration, again I'll draw an image. Let's draw trehalose. Trehalose means 2 alpha glucoses. Okay, so first one like this, again alpha D glucose, so this one is alpha D glucose, and if you add another alpha D glucose, means now just rotate it, okay, so that the first carbon comes this side. So I have rotated it. Now once I have rotated it, just put a special concern here, I have rotated it. Okay, so everything will become flipped. Once I have rotated it, every carbon will become flipped. Okay, flipped in the sense this will remain alpha, but its OH group will come up because it has been rotated. So its OH group has come up. Instead of being down, it has come up. So everything of this, just flip it. Okay, just flip every group. Okay, this is the first carbon. Second carbon, OH was down, so OH will be up. Third carbon, flipped, so H will go up, OH will come down. Fourth carbon, H will come down, OH will come up. Fifth carbon, CH2OH will come down and H will come up. Because of rotation, everything has been flipped. Now again same pattern. Okay, this will be condensation. Once condensation reaction happens, now this is the first carbon again, second, third, fourth, fifth, sorry, this is the fifth carbon, this is the sixth carbon. sixth, second, third, fourth, fifth, sixth. Now look at this linkage 1 to 1 alpha 1 to 1 glycosidic bond. So both the hemiacetal carbons are locked in glycosidic bond and the fourth carbon is free this side. Okay, so means both the hemiacetal carbons are locked in glycosidic bond, so they cannot oxidize anyone, they cannot, you can say, act as reducing agents. Reducing agents means they will oxidize themselves plus reduce others, so they cannot reduce anyone, I should be saying. Okay, so they are known as non-reducing sugars. So this is the concept that you need to know regarding the carbohydrate chapter. 1 to 1 means non-reducing, 1 to 4 means reducing, because the first carbon was free this side. Okay, so this is the only concept that you need to know. Okay. Yes, Amish, I saw the video. I'll just reply to you after this class. Okay, so I hope you are able to comprehend the lecture, and both of you just go through this lecture again. Okay, and if you have had any doubt, just ask me on my personal number. Okay, so that in next class I can again repeat that for you. Okay, then let's meet in the next class. And